Ladies and gentlemen, good afternoon to you all. I am Dr. Subhranshu Sekharkar, Professor of Pediatrics and Associate in Clinical Sciences. On behalf of our Honorable President, Dr. S. Gurumadhava Rao, and our esteemed Dean, Dr. L. welcome you all to today's session of the very informative and up-to-date COVID-19 series of webinars. I will be acting as the moderator for today's session. As you all know, COVID-19 has been ravaging the world since the past several months with devastating consequences. Globally, more than 18 million people have been affected and almost 689,000 lives have been lost. In UAE, as of today, more than 61,000 people have been affected and almost 351 lives have been lost. The great news in UAE is that more than 5 million tests have already been conducted till now, and they are testing more and more. That is why the UAE response to the coronavirus outbreak has been considered as exemplary by experts. Having said that, 2 days back, the WHO says effects of once in a century pandemic will be felt for decades. Last week, we had 3 very interesting talks on infectiousness of COVID-19, decoding the mysteries and debunking of COVID-19 myths. In continuation, today we will be focusing on the diagnostic and pharmacotherapeutic aspects of COVID-19. A recorded version of this webinar along with the presentations will also be made available in our RAC-MHSU website. Before I invite the first speaker, let me clarify some housekeeping issues. The question and answer session will be conducted only at the end of all the 3 presentations. So you are requested to put your doubts in the questions section. The certificate of attendance will also be provided. You are requested to fill up the questionnaire survey, which will be emailed to you. We would love to hear from you in case you have further queries. You can mail us at events@racmhsu.ac.ae for any further clarification. Now I would like to introduce today's first presenter. Dr. Godfrey Menez is currently working as Associate Professor of Medical Microbiology and Immunology at RAC College of Medical Sciences and a clinical microbiologist at RAC Hospital. He received his master's degree in medical microbiology in 2003 from KMC Mangalore and PhD in medical microbiology from JIPMER Pondicherry, India in 2011. He has been a scientist at Erasmus MCA Rotterdam, the Netherlands, and has worked in Hale University, Saudi Arabia. He has set up central research laboratories in a couple of institutes. He has more than 50 research publications in reputed journals and a couple of book chapters to his credit. He is an editorial member of more than a dozen reputed index journals. He will be talking on nuances of COVID-19 diagnosis. Over to you, Dr. Godfrey.
Yeah, sharing a PowerPoint. You able to see the screen? Are they able to see the screen? Are they able to see the screen? Yeah, are you able to see the screen?
Yes, sir, we're able to see, sir. Please continue.
Okay, yeah. All right. Yeah, good afternoon, everyone. Thank you, Dr. Subhranshu, for the warm introduction. I'm here to begin my short lecture on nuances of COVID-19 diagnosis. Yeah, so here you can see in the first slide itself, you can see the transmission electron micrograph of the COVID-19 virus, which is usually shown as colorful, but it is not that colorful. Okay, so the outline of the presentation is going to be review, introduction, diagnostics, when to collect samples, the different molecular tests, antibody and antigen test, other complementary tests, interpretation of the results along with the complementary test, and take-home message. Yeah, but when I do that, I cannot see the screen. Okay. Sorry for the interruption. All right, so we will begin with a couple of questions which are food for thought, or you can say the review sort of questions. Is severity or death toll related to SARS-CoV-2 virus strain? And what is the hallmark of finding in the complete blood count? And what are the requirements, mandatory requirements for specimen packaging? And if you're collecting a single swab, is it a nasopharyngeal swab which is preferred? And what are the biofactory level requirements for carrying out PCR reactions? What are the different indications for carrying out real-time PCR? And interpretations of conformity tests along with the different complementary tests. And why antibody detection tests are not that reliable? What is RT-LAMP? And finally, next generation sequencing in COVID-19 diagnosis. Starting the introduction, COVID-19, that is coronavirus disease 2019, is caused by SARS-CoV-2. As all of us know, it is severe acute respiratory syndrome coronavirus 2. This virus has a single-stranded RNA, which is covered by the nucleic acid, and in turn, it's covered by the envelope. On the envelope, you can find a membrane glycoprotein as well as a spike protein. The ACE2 is the host receptors for the SARS-CoV-2 virus. And this viral spike protein interacts and gain entry into human cells. And a lot has happened since the first case of COVID was reported from China, which was a mystery illness. And you can see that by Jan 10th, within a month, the viral genomes were sequenced and were released, which is crucial for developing molecular tests. And within around 3 months, WHO declared it as pandemic. So it has managed to become a pandemic within around 3 months. Primarily, this COVID is a zoonotic disease, which is transmitted to humans from bat or intermediate host pangolin. Since it happens to be a microbe of a virus of animals, we did not have any clue to begin with about the presentation, disease duration, or the immune response. A few facts about the SARS-CoV-2 or COVID-19. There are incidences of relapse in virus shedding as well as clinical symptoms, and there are also reports of super spreaders where a single index case has led to infection of more than 12 cases. And it is a known fact by now that asymptomatic individuals also are a threat for transmission, and incubation time varies between 3 to 12 days. And globally, we can see that there are multiple strains, different strains of SARS-CoV-2. And primarily the disease pathogenesis is because of autoreactive T cells and cytokine storm, which is triggered by the virus. And more than 80% cases do not need any special kind of treatment, they recover on their own. Okay, so this number has been ever increasing, whereas in UAE, it seems to be under control because of stringent guidelines, and enough of molecular tests being carried out, routinely. All right, so there is a very beautiful initiative by the GSAID, which promotes the rapid sharing of data from all influenza viruses and coronavirus. Daily death tolls are fortunately falling in many countries, and the number of cases, severity, or death toll is not due to viral strains. We initially thought that it was because of different viral strains. Severity and death toll is varying in countries. It is primarily because of depending on how different countries could tackle lockdown or sanitization measures. Okay, as I said, there are different strains, or we could call it as the clade or lineages. Among the lineages, the most common one is the G clade, and a lot of sequencing has been done and it has been uploaded. But these minor mutations do not impact the PCR detection. Coming to the symptoms, the three most common symptoms of COVID are fever, dry cough, and fatigue, which is again variable clinical presentation all over the globe. And no one symptom or set of symptoms can reliably diagnose or exclude COVID-19 infection. So what do you mean by confirmed case and suspected case? A confirmed case is the one which is diagnosed by a gold standard test, that is real-time PCR. Sometimes by virus isolation. We do not prefer virus isolation. It is mainly done for research purpose and not for diagnostic purpose. Whereas suspected case is the one which is having fever with the respiratory symptoms, common symptoms, within 14 days of being in close contact with the confirmed case. So if you have to describe COVID-19 in three simple words, it is multisystem inflammatory syndrome. Okay, hence it requires multidisciplinary approach, people from different disciplines to get together and try to do the needful, with gold standard lab diagnosis being the cornerstone. Public screening in UAE, they've adapted a laser-based DPI and also smart helmet to monitor coronavirus, mainly the temperature rise. Coming to scans like CT scan, no finding can completely rule in or rule out the possibility of COVID. So hence we need laboratory diagnosis. Elevated interleukin-6 and procalcitonin are excellent markers of increased disease severity. And when it comes to hallmark laboratory finding in the complete blood count, the most important one is lymphocytopenia. Along with that, there is decrease in the cell count for leukocytes as well as thrombocytes. All right, and the other markers which are mentioned here are all suggestive of either liver damage, liver injury, or muscle injury, or heart injury, and they are quite significant, though we cannot rely on them totally. A patient need not have all of these laboratory values to be infected. So healthcare people involved in taking care of patients, or involved in specimen collection or processing samples, all of us, it is mandatory that we don PPE, complete PPE, and aerosol generating procedures helps in airborne transmission, usually in the contained environment or in the hospital setup. Diagnostics, to name a couple of them, molecular biology based, which is RT-PCR, serology, which is antigen or antibody based, point of care test, which is bedside test,
Right.
then next generation sequencing, and also other diagnostic tests. Okay, this graph is quite useful. To begin with, SARS-CoV-2 RNA or antigen can be detected even before the onset of symptoms. All right,
Okay.
and then
Right.
till
Right.
7
Last time
days,
I assigned.
the initial phase, where antibodies
Right.
cannot
Right.
be
I
formed,
assigned.
we can still diagnose SARS by using RNA test or antigen test. IgM is the first antibody to be developed, which
If I
develops
am assigning,
starts forming on 7th day and falls by 21st day.
right.
Whereas the IgG production starts by 14th day and it remains for a couple of months. It remains for a couple of months. Okay, which provides long-term immunity. And if you go for test by 14th or 15th day, the test could be positive for all three, RNA or antigen, IgM or IgG. If a single swab is preferred, then it is nasopharyngeal swab, mainly because it is tolerated by the patient and also it can reach the right area of the nasal cavity. So once it reaches the posterior pharyngeal wall, the swab has to be kept there for 10 seconds while being twirled 3 times. And if you're collecting more than one swab, it can be allocated into a same BTM. It need not be 2 different BTMs. And among the swab, the flock swabs are the preferable swabs. And if in severe cases, it's always better we include a lower respiratory tract sample as well. Coming to VTM, it could be in-house or commercially procured viral transport medium. And VTM usually would contain gentamicin to inhibit bacteria and amphotericin B to inhibit the fungi so that only virus remains. Specimen packaging is quite important. The sample containing a receptacle goes into a sealed plastic pouch, which in turn goes into outer packaging. And on the outer packaging, it's very important that we have a biohazard label as well as the complete patient details. So this is mainly done for making sure that there is no spillage, and if at all there is a spillage, it is not hazardous to others. Earlier, it used to be a 2-step PCR where you generate cDNA, and from there you go on for generating double-stranded DNA. But then now we have a single-step quantitative reverse transcription PCR. Starting from RNA, you generate your cDNA and then end up with the cDNA as a template for the process. And this is a beautiful picture of RT-PCR lab, which is a class 2 biosafety level, which is a classical picture of RT-PCR lab how it is supposed to be. It has 4 different sections, which will be dealt in detail by the next speaker. So a direct test versus indirect test. Direct is where you look at RNA or antigen components of the virus, whereas indirect is antibody production or other markers. Screening tests like antigen detection depends on local regulating authorities for their approval. For countries with a larger population, maybe antigen tests are approved tests. We also do have gene expert platform mainly for low and middle income countries, which is quite effective. So what are the different indications for carrying out COVID-19 test? It is useful for confirming suspected cases, deciding on the release of confirmed patients from quarantine, screening asymptomatic individuals in close contact with confirmed case, and also for differential diagnosis from unknown respiratory syndromes. These are different target genes which have been tried out. Okay, the most common ones are E or RDRP, and if it's ORF1, where it doesn't include RDRP, then RDRP will be part of ORF1 sequence. Yeah, so qRT-PCR is the gold standard, and the major advantage is it is used for amplification and analysis, which is done simultaneously. Okay, and both these viruses, SARS-CoV of 2003 and the current SARS-CoV-2, belong to a common subgenus, which is sarbecovirus, and these are the different structural and accessory genes which could be targeted in the RT-PCR. And WHO recommends E genes for screening, whereas RDRP for confirmation. And low CT values indicate high viral load. So low CT values are always bad news, whereas high CT values is the good news. Low CT values indicate high viral loads and may be used as an indicator of transmissibility. So CT value is the intersection between an amplification curve and a threshold line. It is the relative measure of concentration of the target, whereas it is just an indicator. We cannot actually interpret it as viral load. And patients with high CT values more than 35 can be considered non-infectious. And some laboratories, especially in countries with larger population, they have tried out pooling of samples. And if there is a borderline CT values, it has to be interpreted cautiously because it could be false positive or false negative. So retest is always recommended. Regarding antibodies, the antibodies targeted are mainly ones generated against spike proteins and nucleocapsid proteins. And when it's only IgG present, it is an indication of previous infection. So there's a growing demand for rapid point of care testing, and it is useful for surveillance, epidemic forecasting, and determination of immunity, and also useful for public health measures, including return to work protocols and social segregation of susceptible individuals. Yeah, so why antibody detection is not that reliable? At times, IgG seroconversion can happen earlier than IgM, which is rare, but still there are incidences, so that's the reason why seroconversion is a bit confusing. Okay, so when you compare between the RT-PCR and antibody test, so if only PCR positive, it means it is the initial phase, the first 7 days of the infection, whereas if it is positive for all three, PCR, IgM, and IgG, it means the patient is in active phase, maybe the 14th or 15th day of the infection. And if it's only IgG present, then it indicates past infection. Yeah, RT-LAMP is a reverse transcription loop-mediated isothermal amplification. So it has been optimized and SOP is out for SARS-CoV-2. It is capable of being used in place of RT-PCR, and it is as sensitive as RT-PCR, where you do not need too many reagents. It is much faster, and you need not extract RNA. You can start from the sample straight away. Okay, and as I said, in countries with a larger population, they may approve, the government may approve antigen tests for carrying out. So if rapid antigen test is positive, it is to be interpreted as positive, whereas if it is negative, but the patient, if it is symptomatic, then we have to send the sample for RT-PCR. But if the patient is asymptomatic, then it has to be declared as negative, because the countries with larger population, they have to take care of the symptomatic individuals, and they do have enough patients in the hospital admitted in the ICUs, so they cannot be taking bothering too much about asymptomatic individuals. Other tests are like next generation sequencing, which is detecting mutations, which could be used mainly for research purpose, or at times also if it is if a large number of samples can be processed in one go, then it could be useful for diagnosis as well. So sequencing by synthesis as well as long read sequencing. We do have nanopore technology, which is a small compact size instrument, which could be used as a point of care instrument as well. And also of late there are CRISPR based assays for detection of COVID. Take home message: COVID-19 diagnostic uncertainty remains quite a huge problem, and there is urgent need for accurate and rapid diagnosis of SARS-CoV-2 infection and presence of antibodies against SARS-CoV-2. It's a big question whether it correlates with immunity to the virus, and how long the antibodies, IgG antibodies, remain in the bloodstream. So PCR performance is quite robust based on different genes, so that's the reason why it happens to be a good standard. In mild cases, the virus disappears early, so that's the reason why we may miss out on mild cases. And in case of virus in blood and severity, it has been very well correlated. And the key strategy to save lives and protect livelihoods remains testing, tracking, and treating. So I will leave you with this landmark, beautiful landmark of Russell Kaiman. So it's been a common war on the face of enemy, or you can say common enemy, which is a tiny microbe, a virus. So hats off to all the healthcare workers who have been involved tirelessly in this war against COVID. Thank you.
Thank you, Dr. Gottfried, for a very clear and lucid presentation. As already informed, the question and answer session will be conducted only at the end of the 3 presentations. So you are requested to put your doubts in the question section. The next speaker is Dr. Hafiz Ahmad, who is working as an assistant professor of medical microbiology at RAC College of Medical Sciences. He is also working as a clinical microbiologist at RAC Hospital. He did his master's degree in medical microbiology from KMC Manipal in 2005. He has done his PhD from All India Institute of Medical Sciences, New Delhi in 2014. He has also a doctoral fellowship from the prestigious National Institute of Health in Bethesda, USA, and postdoctoral molecular training from Abbott Laboratories, Germany. Dr. Hafiz has more than 30 scientific publications in peer-reviewed journals and generated top-tier funding as co-PI from Jayat Foundation. His topic of presentation today is role of RT-PCR in the diagnosis of COVID-19 infection. The floor is yours, Dr. Hafiz. Dr. Hafiz, are you there?
For the introduction, and thank you, Rakcoms present, sir, respected audience, dear students. I hope you are able to see my presentation. Can you confirm, Dr. Subhanshu?
Yes, yes. It's
All
okay.
right. So, so at the outset, I would just declare that there's no financial or any disclosures. I am in quite involved with RAC Hospital with regards to COVID-19 diagnosis. We, with the initiative of His Highness and thanks to GM Rao sir and Raza sir, they have given us the opportunity to work here and at RAC Hospital and contribute to the UAE for diagnosis and detection of COVID-19 using real-time PCR. So just a brief, Dr. Godfrey has taken a very lucid and elaborate lecture on and made my job quite easy. I will just touch upon the some of the diagnostic parts, especially with respect to the real-time PCR. So we all know that SARS-CoV-2 is it's a family of the coronavirus, and there are different different variants, as Dr. Godfrey rightly mentioned. And the one novel one, what we call as novel coronavirus, also now we call it SARS-CoV-2, is unique because of the ability to cause human to human transmission, which has caused this pandemic. And now we are at a stage where everyone is scared to even walk out of the house. But one thing is good as a virus, because it's an enveloped virus, and a small treatment of soap and water for 20 seconds is enough to to get away with this virus. So as a message goes, wash your hands whenever if possible. If you find that you're touching something, then this is one of the best steps. I would recommend more than hand sanitizer to wash with soap and water, because these are enveloped viruses. And we also know most of the students, we have been teaching them the peculiarity of single-stranded RNA viruses, and what is the difference between a negative sense and a positive sense virus. So these viruses can directly translate, and there is therefore the infectiousness is more unique to this virus. Also, the site of attachment is just like other viruses. It has these ACE receptors, and which can bind to these viruses. And a lot of research is going around to block these receptors, block the viral entry at different levels. And Dr. Ashfaq in the coming webinar would discuss more on these aspects of COVID-19, which is unique to this virus and contributing to this pandemic. For now, for me, I have been given the role for PCR. So all of you must be aware that thanks to this coronavirus, I would say most of the clinicians or physicians know right now what is a PCR, which is called polymerase chain reaction, and we have these steps of sample preparation, RNA extraction, reverse transcription. Dr. Gottfried briefly mentioned many of these. I will just try to go a little bit more in detail of this. So basically, the first step to PCR is something called as nucleic acid extraction. The whole purpose of this process is to concentrate the nucleic acid. So you have a cell, in the cell you have this nucleus, and then you have this nuclear material, which is DNA or RNA, and you have to concentrate it because there's a lot of proteins, a lot of inhibitors, a lot of other unwanted, uh, unwanted substances which need to be concentrated. And if I can highlight using the highlighter, if I can, maybe I can show you with the highlighter. Uh, am I able to highlight this? Okay, I think this is the highlighter. I'm able to highlight the pen. Okay, so anyway, so I think I'm not able to highlight it right now. So let's go ahead with the magnetic based. So now there's a technology most often which is used as magnetic bead based technology for an extraction. So these are small micro beads of magnets, and which aid in the extraction of RNA. So you have a sample of desired volume, and the magnetic beads. So all these kits are commercially available, and then steps 2 to 3 steps of washing of these beads happen before you get your eluted RNA. So this whole process is repeated, and you are able to concentrate the nucleic acids. Because to carry out this PCR, what's called polymerase chain reaction, most of our students, I think, are well aware that Kary Mullis, was the 1985 invented this technology, and thanks to him that we are today what we know of PCR, and he also got a Nobel Prize in 1993 because of this technology. The whole diagnostics, industry has changed. Now the whole industry is moving towards what's called molecular diagnosis, and this is thanks to Kary Mullis. Because the whole process involves 3 steps, because DNA is double stranded. We heat the DNA to 1995 degrees so that it becomes single stranded, breaking the hydrogen bonds, and then steps of annealing. You insert short oligonucleotide primers. Primers are nothing but short stretches of DNA which can attach to these target genes. So if you're targeting COVID-19, you target these specific genes, E gene, N gene, or RDRP gene, and you are able to detect with 100% sensitivity and specificity these nucleic acids, and then you extend the DNA polymerase, especially Taq DNA polymerase. The beauty of Taq DNA polymerase is heat. It is not heat labile. It can tolerate 100 degrees or 95 degrees of temperature, and that's the beauty of PCR. So initially, when PCR was discovered, they used to do this in buckets. Basically, before 1980s, they had 3 different buckets of 3 different temperatures. One temperature of denaturation, 94 degrees, followed by annealing, 54 degrees, and then elongation and at 72 degrees. So they were doing this in 3 different different temperatures. Now the whole process is done by a thermal cycler. A thermal cycler is nothing but a machine which does all these processes very quickly. Within a few seconds, it will reach 94 degrees. The next couple of seconds, it can go to 54 degrees, and then again go to 72 degrees. And this is the beauty of the machine called as thermal cycler, which can do all these procedure of denaturation, annealing, and elongation in one cycle. So this is called one cycle of PCR: denaturation, double stranded DNA, and anneal it and then extend it using the Taq DNA polymerase, using buffers and this coenzymes, magnesium chloride, and other ingredients, which is called a cocktail. Cocktail is nothing but a master mix. Because these things are required to carry out the reaction, and any of these, if there is a if there's a dearth of any of these molecules, the reaction will stop. So you have to supply your reaction with a template. The template is your sample. It's a COVID-19 sample, nasopharyngeal swab or oral throat swab, and then you have these, the DNA molecule, you Taq polymerase, DNTPs, magnesium chloride, and all this together. And of course, you have to have a detection technology, which can be either a primer, a probe, or you can have a gel electrophoresis. So in traditional PCR, what we have, this is the machine. What I'm trying to refer is the thermal cycler. In fact, Central Research Laboratory at Rakcoms HSU, we have the some thermal cycler placed, and we can do the reaction in these mini small ampules, which is like 100 to 200 microliters. 1 microliter is 1,000 of a ml. So imagine the low volumes we are, therefore precision pipetting or a good neat hands are very essential to do a PCR reaction. On the contrary, in biochemistry or in others, the micro level pipetting may not be that necessary, but when here you load 1 microliter of Taq polymerase per reaction, 2 microliters of internal control, and then therefore the chances of error are very high. And therefore, this PCR, the beauty of PCR, that it can amplify. It's nothing but an in vitro amplification of DNA. So even the smallest virus particle, if it's there in your body, it will be amplified and it can be detected using this technology. And then these are loaded on these machines, and then you can detect them either using agarose gel, which is the traditional electrophoresis gel. You have DNA, because it's negative charge, migrates towards the positive charge, and it can be detected with based on the charge mass ratio. It gives a pattern, and the pattern can be detected and matched to different diseases. This is the conventional PCR, what's called as polymerase chain reaction. This is how, in this again, this is placed in our Central Research Laboratory, and we are doing a lot of student research projects, thanks to thanks to Rao sir and Dr. Ashfaq. We have been able to do a lot of undergraduate research with this technology. Now, coming to the difference between a traditional PCR and a real-time PCR, what Dr. Gottfried has already mentioned. The unique difference is, of course, with the real-time PCR, you have these probes, the probes which can attach to your DNA, and they, with each cycle of denaturation, 95 degrees and annealing, they excite a fluorescence, and that fluorescence is detected by the camera, and that's what's called as real-time PCR. It's also called as RT-PCR if it's a reverse transcription, like in this case of COVID-19, it's an RNA virus. So you reverse transcribe cDNA to DNA, and then you amplify using these using primer and probes. It's also called as qPCR. But these are all slight differences in the names. qPCR refers to quantitative PCR. A quantitative PCR will tell you how much of virus is in the body based on the CT values, or based on the how much of copies per ml of viral load is there in the person's body. Whereas a qualitative PCR can just tell you, yes, he's positive for COVID-19. No, he's not positive for COVID-19. So that's the slight difference between qualitative and quantitative PCR. So we have different dye sets. One of them is the cyber green dye, which is again an intercalating dye, and it can intercalate between the double strands of DNA and gives a fluorescence. And this fluorescence is catched by the camera, and then in each cycle of PCR amplification, the camera catches, and how much of based on the amount of nucleic acid, you are able to detect how much of virus is in the body of the person. So this is there are other technologies which currently used right now. What we are doing with PCR is the Stackman probe technology, which is very important with respect to real-time PCR detection. So you have a forward primer which you have designed based on the COVID-19 strain, which was originally from Wuhan. You have a reverse primer, and these primers attach and sit on the sample's nucleic acid, and if it matches, they extend based on the DNA polymerase, and they're able to cleave this the moiety, or what's called fluorophore and quencher. The beauty of this TACMAN PCR is that if these two are together, the fluorescence and the quencher, they do not give a signal. But once if you have the target molecule, or if you have COVID-19 presence, then the DNA polymerase will extend and will cleave this molecule, and it will excite and give signal, and you will get fluorescence products which are captured by the camera, and this gives you the detection of the person is positive for COVID-19. So different companies play around with different technologies, but this is TACMAN is one of the best, Raj. About many companies which are in the market, they only go with TACMAN probes, and because of the concept of a CD value. So the DNA amplifies, and there's a curve, a sigmoid curve is seen, because as the DNA amplifies, it gives this peculiar graph, and the graph is plotted in the table. This actually is a sigmoid curve, but when you see in this window the fluorescence versus cycle, so as the amount of DNA increases, the lesser the CD value. And this is what this graph, this next slide shows the concept of the real-time PCR. If a person has a CD value of less than 12 or 15, than a person who has a CD value of 24 or 30s, then the lower the CD value, the higher the viral load, COVID viral load. And now a lot of research is coming, and we will discuss in the coming slides. So the higher concentration of virus, the lesser the viral load. So there's an inverse relationship of fluorescence versus cycle number, and this is the only thing you need to remember for real-time PCR monitoring. So we already mentioned these are the thermal cyclers which can detect, and as there's a background signal always. The plastics, the different dyes, they have some signal in them, but these are normalized. So normally the instrument normalizes these signals and gives you a curve, something. So there's a threshold. If only above this threshold, if there's a signal, then only the machine will detect, and then only the patient is called as positive sample. So this is the concept of cycle threshold and the concept of baselining the data. So with respect to fluorescence and cycle numbers, you can detect your amplification in real time, and that's the name comes from real time PCR. Every cycle there's a fluorescence, so we go up from 1 cycle to 35 to 40 cycles, and we try to analyze how much of virus is present in the individual. This is again one of the real-time PCRs in our central research laboratory, and alhamdulillah, we are doing a lot of research projects with these instruments and the CRL. Coming to the more core genes involved that Gottfried mentioned. So genes, we have these RERP gene, the E gene, the N gene, and these are so different countries have different protocols for detection. So normally E gene, what we see is seen in most of the first gene to appear is the E gene when we see the viral dynamics, and then the next one is N gene, and then the last one to appear is the RDRP gene as the viral. So the E gene is used as a screening assay. As a screening tool, we use E gene, and we confirm by using the RDRP. RDRP is the polymerase gene, RNA dependent RNA polymerase gene, and then confirmed by the. So I'll not go into too much detail, but just to give you an idea, this is the genome size, genome structure, and these are the detection genes which are used in diagnosis of a PCR for COVID-19. This is one of the snapshots that we took from the lab, and it shows you how the interpretation of samples are coded, and you can get positive for all the 3 genes, you can get positive for 1 gene or 2 gene, and the curves are seen in the instrument, and you are able to diagnose the person with confidence that this person is positive or negative for COVID-19. So there's a lot of commercial aspect going with COVID-19. Hundreds of companies have pitched in for selling their COVID-19 kits. Some are good, some are under evaluation. But the only message I have for the laboratories is you make sure you validate your results, be it a real-time PCR, or be it a LAMP. LAMP is not so much commercially used, but if you are going ahead with it, you make sure you validate your results, because a lot of there's a lot of false negatives. So imagine the sensitivity of an nasopharyngeal swab is 60 to 70%. So you're already missing 30%. And if you have a technology which is not good enough, you're missing 50% of the cases. So there is a lot of the PCR diagnosis it has to be very closely monitored. Which system is being used, because there can be open system. People are coming up with their own protocols. Also, closed systems. So closed systems have the advantage that everything is prepared and commercially available and CE approved. Many of these kits are already commercialized. Approvals are given, and you can go ahead and purchase them in the market. And there's no need for too much of R&D to be done from your side, because it's a patient's life. At the end of the day, you are giving someone an isolation for 1 to 2 months just because you're falsely detecting it, which is never appropriate. And so therefore we as far as as the COVID-19 diagnosis goes, we make it very sure that we follow the right protocols and make sure that the patient is getting the right reports. So we prefer automated PCRs, automated isolation techniques, automated, so there's less of manual involvement. And because now we're talking about samples, Dr. Subranju mentioned almost so millions of tests are already done. 500 tests already done by UAE, and therefore it is important that these processes are automated and a flow is already maintained, so that you have less chances of errors. And that's why, thanks to the initiative by UAE, that we are able to do a lot of these tests because of these automated processes. So this is a machine which is automated. It does the extraction on the master mix together, and then you detect using this PCR. And there's no manual intervention, very little manual intervention, only sample preparation. Sample can be loaded here, and you can get the results here. So we are going towards Latifa, the DHA, and Abu Dhabi. They all go with mostly automated protocols. So as I mentioned, the molecular diagnosis, the key is that you need to make sure that the interpretation is very correct. So someone has to watch these curves, watch these runs, and validate them before it goes in to the system and gets validated by the doctor, because there can be a lot of errors. So internal control is one moiety which gives you an idea, because it runs, it's a control which is designed by, which is sometimes plasmid or sometimes housekeeping gene, which is conserved, and it does not change based on the patient parameters or other parameters. And this is an example of a bad internal control. You see, these are 96 samples, and the internal control is supposed to give you a very tight band, or something like this, which is going, crossing the threshold, crossing the CD cycle, and giving you a fluorescence. But if you have internal control like this, then you cannot rely on your results. So this is just a little bit more technical thing which I'm trying to discuss, but it's a very important thing that internal control has to be run with each PCR, and positive control and non-negative control, and then internal control. Then only you can release the patient reports with confidence, and that's what we're doing. So other thing which is very important is that there has to be a unidirectional flow of PCR. You cannot do everything in one room. First, you have to have a room of sample preparation or RNA extraction, followed by a master mix room, and then the PCR room. So it has to be compartmentalized with respect to air, as well as also negative pressure needs to be there, or biosafety level 2, as mentioned by the speaker. This is important so that there's protection. Yes.
Hafiz, you have got 2 minutes left.
Thank you so much. So I'll just so again, Dr. Godfrey already mentioned the swabs which is used. I will not go into main detail. Uh, so one thing what I want to mention here is the dynamics of viral loads are very different. The same virus you may be able to pick 100% when in week 1, the same test, the same kit, but you in the week 3, we will not be. So the timing of the sample is very crucial, because some patients may shed the virus more than asymptomatics who may shed the virus less. So therefore, it's very important. There are different algorithms for symptomatic patients, for asymptomatic patients. But you need to know that there are certain set of guidelines which are set for detection, and DHA has also given some guidelines. These are some of the guidelines from some of the kit manufacturers we have. If you're able to detect all, you say positive. If you're detecting only one, you say presumptive. So these are some of the nuances or details of diagnosis. One important thing I want to mention is the limit of detection. So in asymptomatic people, now we have mass of people who are asymptomatic without symptoms, and there's no guideline for that. So therefore, it's very important to do sampling for people who also who are asymptomatic, and now the government is slowly heading for it, because we need to screen all the people irrespective of the symptoms, and then only we can be sure that the country is free of the virus. And so there are some also considerations of calculating viral loads based on the CD value, because CD value is inversely related to the viral load, and it can help people to make decisions. So sometimes clinicians, will tell you verbally that what is your viral load. Uh, Again, another thing is sometimes people think, why I'm asking for test, why is retesting? But retesting is always important, because sometimes you are not in that phase of the virus, the viral dynamics. So therefore you have to get a test again because of some inhibitors in the PCR, or because of some interpretation issues. A new test, a new sample may give you the value and can get you a confirmed result. So don't think that it's just that retesting is it is essential thing. One thing for all real-time PCRs is RNA virus. RNA is, we our hands are, the RNAs is there in the environment. So therefore, you have to wear all the PPE with the gloves, non-powder gloves, because powder also, can cause inhibition. And some of these things like, sample inhibitors, or VTM issues, or if you don't clean the labs, you may have contamination. And therefore it's very important to clean or with bleach and other things to just avoid contamination. Just some thoughts for the day, which there's some new concepts are coming because of viral load. Just viral load predicts the mortality? Just viral load? Yes, it does. So if you have a viral load which is very high, less than CD is less than 24, some people say that you can correlate more of the prognosis if by saying CD value. Or is it a true value, or is it an instrument issue? These are some of the burning questions which people have. Am I infectious if my CD is more than 24? Because the virus is different. The virus which is in the US is little different from the virus which is circulating here, the strains, and therefore these cannot be changed. There may be a publication on the net which tells you, but unless there is a research which is done here, you may not get your cutoff. So this is the hospital lab, and I'm thankful that my Dr. Godfred, Dr. Pooja, some of the university colleagues are contributing to this. Thank you so much for your patient hearing. Any Dr. Subhranshu?
Yeah. Thank you, Dr. Hafiz, for a nice and exhaustive presentation. You made a complex concept very simple to understand. Now, the last but not the least panelist for today's webinar is Dr. Suresh Kumar, who is working as Assistant Professor of Pharmacology in Rakcoms, MHSU. He has completed his basic medical degree, MBBS, from RGUHS Bangalore, India, in the year 2005. He has done MD Pharmacology in 2011 and DM Clinical Pharmacology in 2014 from JIPMER, Pondicherry, India. He has been involved in teaching and research since then. He has published 25 research articles in both national and international journals. Several book chapters have also been published. He will be talking on pharmacotherapy for COVID-19. Where do we stand? Over to you, Dr. Suresh.
Yeah. Thank you, sir, for the kind introduction. Good evening, everyone. So after knowing everything about the RT-PCR and the criteria of diagnosing the COVID-19, we shall see what are the current available options as far as the treatment is concerned in the next 20 minutes. Yeah, so this is the outline of my talk. So we shall see what happens after the infection, after the virus gets into the body, and how do the symptomatic COVID-19 people present. This is the next thing that we'll see. And what happens after testing? Say a person is tested positive, what is the next flowchart that we have to see? And what should I do if I develop symptoms? And the current pharmacotherapies that are available, and what are the investigational drugs that we have, and also the latest UAE national guidelines for the management of COVID-19. So this is a very important basic slide which tells us the infection happens at the 3 stages within the human body. The first stage is the early infection. The second stage is at the pulmonary level. The lungs will be affected. The third stage is the systemic disease. Systemic disease involving each and every organ of the body. So the infection has spread throughout the body, and more than infection, it is the inflammatory response, the immune response of the body to the virus, which has exaggerated, and that is why we see the involvement of the lungs, heart, brain, and kidneys. And there is coagulation, there is inflammation, which is referred as thromboinflammation, and there is vascular disease and cytokine storm. So, yeah. So once the virus gets in contact with the alveolar cell, this is the alveolar cell within the alveolar space of the lung. So it utilizes this receptor, the ACE2 receptor, and then enter into the cell. And this process is called as endocytosis, which is facilitated by other proteins also. There are some drugs which can target this process. Once inside the cell, the virus will be within the endosome, which is then released from the endosome as a single strand RNA. This single strand RNA get replicated with the help of this enzyme called RNA dependent RNA polymerase into multiple copies. So this is the key enzyme which is responsible for enormous multiplication of the viral copies within the alveolar cell. So then we have the protein synthesis also on the other hand, and ultimately the virus is formed, and then there is exocytosis, and this new virus released from this cell is able to infect the another cell, and eventually all the cells will get destroyed, and that leads to the fluid accumulation within the lung space. So now we'll see how the body will respond. So initially the macrophage will be the one to respond, and then the neutrophils, and they start secreting the cytokines. Okay, mainly these are the 2 cytokines which is shown to be important, the interleukin 6 and interleukin 1 beta and interleukin 8, and this leads to the accumulation of fluid, and that hampers the gas exchange. That is the reason why people go for hypoxia, because of excess of fluid accumulation within the lungs, and then eventually the alveolar collapse, and in severe cases people go for acute respiratory distress syndrome, systemic inflammatory response syndrome, thromboinflammation, septic shock, and multi-organ failure. Now we'll see what are the targets where the drugs can be used. So we can target the entry level with the help of this drug called camostat, which can inhibit the entry. Then chloroquine under investigation. Omifenovir is also another drug which is tried for the inhibiting this particular path of viral entry. Then we have this enzyme that is RNA dependent RNA polymerase. This can be inhibited by these drugs, remdesivir, favipiravir, and ribavirin. And then we have the protease inhibitors, and then we can target the antigens secreted from the viruses with the help of the antibody, with the help of the convalescent plasma. And interferons are the natural defense mechanism of the body. Interferons are known to cause apoptosis, the programmed cell death of the alveolar cell, which is said to be very, very beneficial. But we need more data on that as well. And we can target the interleukin 6 with the help of the monoclonal antibody, anti-interleukin 6, and then the inflammation, which can be suppressed by the corticosteroids, which are shown to be very promising in the management of critical cases. So these are the symptoms and the features. So just I'll concentrate on the hypoxia part, because it can start even in the lung phase, the pulmonary phase itself, it can start. And other markers, abnormal chest X-ray could be seen, and mainly the inflammatory markers will be elevated enormously in the critical stage of hyperinflammation. So what happens after testing? Okay, say a person is tested positive by the RT-PCR. So all positive patients should be going for isolation. So it is a very important aspect, very important measure which should be strictly followed in the view of public health, because each person can be transmitting if he is not isolated to many other people. It could be at home or the isolation facility or even hospital, that is based on the individual case scenario. And the test will be repeated, RT-PCR will be repeated on the 10th and the 12th day, and after every 24 hours after that, after the 12th day, until the person shows no symptoms or the 2 consecutive samples are taken, are shown to be negative, When this is fulfilled, then you can say the isolation ends, but has to follow the universal precautions of social distancing. If the person shows negative, still he will be, because he has the history of contact, he will be under 14 days of quarantine. And during the quarantine period, if he develops no symptoms, he will be resuming the normal work, maintaining all the measures of social distancing, mask and hygiene practices. If he shows symptoms within 14 days, he has to go for the test again and follow the same flow chart. What should I do if I develop symptoms? So the first thing is, importantly, we have to isolate ourselves, put on the mask. It is very important. It's the responsibility that we should not be the cause for the infection for others, and it should be duly informed to the single point of contact so that the health authorities are notified, and we should isolate ourselves. And very important, do not panic. These are the hotlines available in our country for health assistance in case of development of symptoms. Current status. So we would have heard by this time that there are no FDA approved drugs for the treatment of COVID-19. Also, as per UAE COVID guideline, there are no approved therapy for COVID to date. But is it all that bad? Okay, we'll see what the statistics says. This is the global COVID-19 statistics as of today, that is from the Worldometer. So we have the total cases of 1.8 crores. Of them, 62% of these people have recovered. 62% of these people have recovered, and 33% are the active cases, and deaths have been 3.8%. 3.8%. Of these active cases, you could see here, of these active cases, 98%, almost 98% are mild cases. That means they are still in the stage 1 or in the partial of stage 2, but definitely not critical. But only 1% would be accounting for the critical cases. That means body, the immunity itself is able to fight against the virus very successfully, so that among the active cases, only 1% will land up in the critical phase. So that's a good news, because just because there are no specific therapy, it doesn't mean that all people should get panicked. So the specific medications that target the pathway, which I was referring in the earlier slide, the antiviral drugs, which are the specific medications, could be used off-label or compassionate use, or as a basis of experimental use based on the available literature, because the disease literature is only from the last 6 to 7 months. So based on that literature, we can choose these antivirals for compassionate use. And that is also again on the case-by-case scenario. So what is this referred to? The investigational new drug, IND is the abbreviation commonly used. It's nothing but the experimental drug. Okay? So IND is any pharmaceutical product being tested for the safety and efficacy in a clinical trial. But any drug is still considered new, even if it is used for many years. Say if one drug is there which has been used for many years, but it would be considered new if it is used for the new indication, or if you change the formulation. Until now it was available as a tablet, now you want to change it as the spacer. The change in formulation or change in route or use in the different patient population. Earlier the drug was used in adults, now you want to use it in the children. then it is considered as a new indication, new patient population, and change in packaging. Okay. So as in this case of COVID, we have been repurposing the already available drugs which were used for influenza and been tried for the COVID-19. That's how it becomes a new indication. That's why all these drugs are considered as experimental drugs. Okay. And unapproved use of an approved indication. Unapproved use of an approved indication is also commonly referred as off-label use. As in case of the same scenario, the anti-influenza drugs, if it is tried for COVID, it becomes an unapproved use, which of an approved drug which is already in the market. So we call it as off-label use. So these are the conditions. Then we can use compassionate use or emergency restricted use or temporary authorization for use. All mean the same. These are the conditions. There should be no proven effective treatment existing for the disease, as in the case of COVID-19. And it is not possible to initiate the clinical studies immediately, exactly because we are in the health emergency. Data providing preliminary support of the interventions, efficacy and safety are available, at least from the laboratory or the animal studies. So if you have at least some evidence of laboratory of animal studies, we could think of using it for the compassionate use, especially for the critical stage patients. And it is suggested by appropriately qualified scientific committee and also drug regulatory authorities of the particular country. And this is based on the risk-benefit ratio, And the next condition is adequate resources are available to ensure the risks can be minimized. That is why we can use these drugs preferably in the hospital scenario where you have the facilities of tackling any adverse reactions or any other risk arising by the use of these drugs. And patient informed consent is very much required. The doctor need to discuss with the patient or his attenders before he gives these experimental drugs for compassionate use. And emergency use of the intervention is monitored and the results are documented and shared. This is very important because we need to develop the clinical data as far as the safety and efficacy of the drugs that have been used currently for the coming cases of COVID-19. So this is the latest guideline from UAE, the National Guidelines for Clinical Management and Treatment of COVID-19 4.1 version. And this guideline is not a substitute for good clinical practice, and clinical judgment based on risk and benefit is needed for individual cases, and it has to be taken in individual decisions. These are the overview of all the available therapies as of now. So we can divide it into the two broad groups. One is symptomatic and supportive group. The other one is the specific treatment, which are mainly the antiviral drugs. Symptomatic, if the person is having fever, we can give antipyretic that will bring down the fever, like paracetamol. For body aches, we can give analgesics, mainly for the milder cases of COVID-19. Hypoxia, we have to give supplemental oxygen. That can be to the extent of non-invasive ventilation, mechanical ventilation, or even ECMO, that is extracorporeal membrane oxygenation. In case of bacterial secondary infections, we can give antibiotics. Coagulation, we may have to use anticoagulants. And immunomodulators, which will target mainly the cytokine pathway. So there we can give corticosteroids, which are very good anti-inflammatory. The monoclonal antibodies, we can target the interleukin-6, that is tocilizumab, interferons, and then we have the convalescent plasma therapy, the chloroquine, hydroxychloroquine, azithromycin. All these drugs are repurposing. We are trying it in the COVID, which are already there in the market. These are all the repurposing drugs, but the data is still has to be more evident. Omifenovir, favipiravir, is no specific treatment needed. There is no specific treatment needed if there is no symptoms. But for high-risk people, only for high-risk people, the doctor can prescribe the compassionate use of chloroquine phosphate and hydroxychloroquine, which is claimed to be having some role in preventing the viral entry within the alveolar cell. And if the person develops pneumonia, you have to follow the pneumonia guideline, which will be the next slide. These are the people who are considered as high risk because the mortality is high in these people, age more than 60 years, cardiovascular disease, hypertension, diabetes, and pre-existing lung disease, immunocompromised, the people with renal transplantation, immunosuppressants, cancer patients, and obesity with body mass index more than 30. When to seek urgent care? Okay. So these are the symptoms of people who are in the home isolation. These are the symptoms they should be very carefully looking for the indication for reaching the hospital very quickly. Difficulty in breathing. These 4 categories. And these are the emergency signs: obstructed or absent breathing, severe respiratory distress, central cyanosis, shock, coma, and or convulsions. So these people should be having the immediate access to the emergency airway management to ensuring the patency of the airway and oxygen therapy during resuscitation, so that to target the saturation of more than 94%. And these are the compassionate use drugs available for the severe patients, that is critical patients. This is the 4th scenario. So here the drugs are given up to 10 days, but here the favipiravir and camostat can be combined with nebulized interferon alpha, which is usually given by the ultrasonic nebulizer, lopinavir, ritonavir, interferon alpha 2b and interferon beta 1b. The other combination is remdesivir, which can be given parenterally up to 10 days. And because the critical patients are already in the stage of thromboinflammation, so antithrombolytics or anticoagulants have a definite role. Anticoagulants have a definite role, and corticosteroids to suppress the inflammation. And to target the interleukins, we have tocilizumab, which can be used to suppress the cytokine storm, and that could be done with consultation of a rheumatologist. And convalescent plasma to be considered as an experimental therapy only in the context of clinical trial, And some devices like extracorporeal blood purification therapy, which will specifically remove the endotoxins and cytokines, especially in cytokine storm. Such devices also can be used only in this context of clinical trials. They can't be used otherwise. And corticosteroid is very promising. Of all the available drugs that have been enlisted, they have shown a good result in improving the, bettering the mortality. So the COVID guideline recommends low dose early corticosteroid in moderate to severe COVID-19 pneumonia on case by case basis. So as the person requires supplemental oxygen, it is advocated to start with corticosteroids. And CDC also recommends the use of dexamethasone in patients who require oxygen therapy. The other option, mainly to improve the oxygenation, is the CARP protocol, that is COVID awake repositioning proning protocol. And this protocol is changing the position for 30 minutes to 2 hours in all the 4 positions so that the oxygenation is improved. This is also shown to be, the early data says it has good effect in improving the oxygenation in critical patients. And this is the summary slide.
Yes, you have 1 minute left.
Yes, sir. This is the summary slide, the 4 scenarios and the respective drugs, which is shown in the boxes for compassionate use. And then we have the supportive care drugs, starting from the antipyretics, analgesics, antibiotics if needed, anticoagulants, oxygen, corticosteroids, tocilizumab, non-invasive ventilation, invasive ventilation, and ECMO. Of course, all of them will be under isolation. So to summarize, the specific drugs are being used as emergency restricted use. So the emerging data also suggests we know about the disease and the drugs being used better than we were 6 months before. Still, we need more evidence for the safety and efficacy. And as Sir William Osler says, medicine is a science of uncertainty and an art of probability. Only if we walk the path, we will know more about the disease and the drugs that we have been using. So that is possible only with the help of the frontline healthcare professionals. So tributes to them, and also the patients who endured the infection and unfortunately who have succumbed to the infection also. So until then, we have to strictly follow the precautions of social distancing, mask and hygiene practices. Thank you for kind attention. Thank you, sir.
Thank you, Dr. Suresh, for the very clear and informative talk. Now we'll start the question and answer session. I request all the panelists to switch on their webcams and be available. Before we start the question and answer session, I just want to emphasize one fact. For those watching this webinar, especially the medical students, you can always refer to the national guideline for clinical management and treatment of COVID-19. It is regularly updated, and the last update came on 15th of July 2020, version 4.3. It is prepared and reviewed by the National Clinical Committee for COVID-19 Management and is endorsed by the Ministry of Health and Prevention, Dubai Health Authority, as well as all these hospitals inside UAE. Now coming back to the question and answer session. we have almost 9 questions here. And the first question I'm going to ask is to Dr. Godfrey. What is the difference between strains and mutation?
Okay, yeah,
It is asked by one of the students.
It's got a basic question, but interesting question. Yeah, it's not related terms. A strain is a microbe when you fully characterize it. If you talk about the terms of bacteria, biochemically and genetically characterized, then you call it as a strain. If it's a virus, then you know the complete genetic makeup of the virus, then it's called as a strain. Otherwise, we do not call it as a strain. Okay, mutation, of course, it can be a point mutation or missense mutation. It's because of exposure to radiation or while copying cell division, they can be errors. So that can lead to mutation. So that's the difference between the strain and mutation. So it's not related terms. But anyway, I hope I answered the question.
Right, I think he has got that. The second one, this is for Dr. Suresh. What does the meaning of cytokine? What is the meaning of cytokine storm? So this is, I think you have already explained in your slide, but again you can tell again so that the student knows
storm
about
is
it.
excessive. Cytokine storm is excessive release of cytokines, which are the proteins secreted by the defense cells of the body, that is the macrophages and the lymphocytes, neutrophils. So they are the response, the immune response of the body. If it gets exaggerated, we call it as cytokine storm. So it is so much more that the inflammation will be triggered. So if the inflammation cascade gets triggered, you could see the thromboinflammation throughout the body. So wherever throughout the body, you can see the coagulation, that is the blood gets clotted, and also the inflammation could be seen. So this will be a more problematic because there will be multi-organ failure. So you can't only concentrate on the lungs, there will be multi-organ failure, and the prognosis will be very difficult in such cases.
Yeah, perfect. So in simpler terms, cytokine storm can be easily defined as an immune dysregulation where a lot of cytokines get released very quickly. That should suffice, right?
Yeah.
Okay. Now the next question is for Dr. Godfrey again. How long these antibodies would be effective in our body?
Okay, yeah, excellent question, $1 million question. Those who are interested in vaccine, this is a very important question. So as of now, the antibodies have been demonstrated to stay in the bloodstream for almost like 5 months as of now. We have to wait for some more time, maybe next year or after 6 months, if we do some seroepidemiology, we are going to know much more about it. And it's a big question whether antibodies, whether they do give us long-term immunity. So it's a big question, and if it is so, it is an excellent remark for vaccine production. Otherwise, for vaccine production, it's going to be a major drawback. As of now, 4 to 5 months,
Right.
Based on literature, antibodies have been demonstrated.
And the literature is rapidly evolving also, so we'll get better answer maybe subsequently within a month or two.
Exactly.
Anyway,
exactly.
The next question is again to you, to Dr. Godfrey. How efficient is DPI test when compared to RT-PCR? Is it reliable and a better alternative than RT-PCR?
Yeah, so DPI is optical based modulation. It is just a test. In UAE, we are always happy to have launch something for the first time and say we have launched it for the first time. So it's more like a screening test, non-invasive, but it's not comparable to any of the laboratory tests like RT-PCR. It has not been compared. It is not comparable, so it cannot be sort of confirmatory test. It can be just a screening test, just like these thermal cameras. Just to screen, okay, but nowhere it could be used as more than a screening test. EPI versus, I guess, RT-PCR. Was that the question?
Yeah, that's
Yeah.
the answer that you
Okay.
told.
Okay. This is
I think that's clear.
nowhere comparable. Yeah. Yeah.
Okay, now this question is for Dr. Hafiz. What is the difference between quarantine and isolation?
So quarantine basically is both ways. You have to, the person has to keep himself away from his relatives or I mean meeting anyone in the vicinity, and kind of lock himself up. But isolation is more of a medical term which is used in hospitals, where you have where you are in the supervision. I think this is maybe Dr. Suresh was mentioning that, and maybe he can take forward my question.
Yeah, it's Dr. Suresh, you
Exactly.
can elaborate a little bit.
Yeah, exactly. As Dr. Hafiz told, the intention is the same. Both the scenarios, the intention is the same. The scenario is different. Quarantine is a person for the person who is just a suspect. Tomorrow he may be negative. We don't know. We are just keeping him in a waiting period of 14 days, so that in 14 days he may develop symptoms or he may turn to be positive, so that we do not be spreading the infection. On the other hand, isolation, we have a person who is already proven to be positive, so you should be more careful with people with positive, and that usually happens because positive people usually will be in the hospital. So this scenario is different, but the intention is the same. That we are not allowing the infection to spread.
Hope that's clear. Now, next, again, the question is for Dr. Hafiz.
Yes, sir.
How long does it take for PCR to show the result? Are there any other more sensitive and faster tests for that regard?
Yeah, that's a very nice question. So normally we with the PCR cycle is around for 5 to 6 hours we are able to get the results. But sometimes also due to repeats, we say what's called turnaround time. We give the patients the turnaround time for 24 hours or longer, because sometimes the repeated testing is required. That being said, also there are new PCRs coming in the market which can detect in 4 hours time. But again, it's up to validation and a lot of LAMP. Dr. got mentioned one of the tests. It can give faster, even faster, maybe in 3 hours time. Even now, there's a proposal going for airport screening to using some of these rapid technologies of PCR. They call us point of care PCR tests, which can give results within an hour's time or within 2 hours, because the patient cannot wait in the airport for 3 hours or 4 hours, which is minimum, because the amplification will take minimum 1.5 to 2 hours. Extraction will take 1 hour, and again sample preparation. So it's a 4 to 5 hour cycle, unless you use something novel technology to overcome this time. Hope this
Right.
answers.
And now you can also take the other question which is related to that. I think you have also mentioned this answer during your presentation. Any cross positivity of PCR with other coronavirus? If
So
it is there,
again,
then you can always.
so what when the tests are in the market, out in the market, especially these approved tests, they do validation with 45 other viruses, like, because these viruses are very genetically similar, than the concept of strains. They're very much only maybe few nucleotide differences will change the target. So therefore, when the kits come in the market, they are validated with other respiratory viruses, even other coronaviruses. It's not necessarily a novel coronavirus, but there are other coronaviruses which may not cause any infection or any person-to-person transmission, and therefore all these kits are basically tested on 40 to 50 different respiratory viruses. So when this test was initially evolved, the clinicians first did a respiratory panel to rule out other respiratory viruses which are similar to coronaviruses, and then when the suspicion aroused in the early 2000 late December or early January, then they came up, the PCR was developed in Jan, and then they came up with this PCR testing. So these tests, there is cross reactivity, but if with the approved test, it is already validated, you do not worry. But if you're doing a homegrown test, you have to validate with other respiratory viruses.
Okay. Now this question is for Dr. Gottfried. Some COVID tests become positive even when the virus is dead but circulating in blood. So what is the test by which we can definitely confirm that the person is COVID negative and the virus has passed or died after the infection is over? And how long does it take for the result to be released? Yes, Dr. Godfrey.
Yeah, very good question there. Yeah, so RT-PCR detects the viral RNA, but it is not for sure that virus is still alive. So that's where the drawback is. It's a gold standard. RT-PCR is a gold standard. It's a quite a sensitive and a specific method. But then the drawback is that it can pick the RNA from the virus which is dead. RNA tends to remain for a couple of weeks, maybe up to 4 to 5 weeks even after recovery. As of now, since antibody test is also complementary test to RT-PCR, along with complementary test, it will give you an idea whether it's acute infection or a past infection, and that is good enough. And if you are, research-wise, if you take a step forward, then you can always try to grow the virus, and it's possible to grow the virus. And if it does grow, then that means it is a live virus. And second part of the question was, was there a second part of the question in that?
The second part of the question is, yeah. So
How long it
which
takes?
test can definitely confirm that the person is COVID negative
Okay.
after the virus has, died?
Okay. No, I think the second question is more related regarding the gold standard test, confirmatory test. So nothing to beat RT-PCR or nothing comparable with RT-PCR as of now. Though RT-LAMP, which is reverse transcription LAMP test, or certain antigen tests could be useful, but then it has to be approved by the local government authorities. All right, and when you have multiplexed PCR, when you have 3 different genes targeted in a PCR, for example, if it is RDRP and E gene positive, then it is less likely that it is false positive or detecting one of the other coronaviruses. But if it's only N gene positive, but it is negative for RDRP and E gene, then the PCR interpretation itself says it could be positive for SARS-CoV virus, which is one of the coronaviruses, but not COVID.
Okay.
Yeah. So,
now, now I will just ask Dr. Suresh to comment on the second test to check the negativity status. Is it advisable or not?
Second test, sir.
Yeah. So the thing is, the government of Dubai has stopped doing a second test to see if a COVID positive asymptomatic person has become negative before releasing them from the 14 days home quarantine. So your comments on that, then probably even Dr. Hafiz can contribute to that. So now what they are doing is, initially when someone is asymptomatic but COVID positive, so they are releasing after 14 days if they are symptomatically recovered, or even there is no symptoms also. Yeah. And there is no need for a second test, so they are not testing them again. So what is your take on that? So should it be tested, or the strategy is correct?
Because RT-PCR is the gold standard, ideally it is advisable to test and then release.
So just to take on Dr. Suresh's question, there is a discrepancy in the guidelines, DHA and DOH and MOH. So with respect to DOH, they say 2 repeat tests is required, and with respect to MOH, but let's go clinically with the dynamics of the virus. The symptoms, you get the onset of symptoms after 5 to 6 days, and then there's a peak period in 10 to 12 days, and then after 14 days you are almost asymptomatic when we talk about symptomatic people, and the viral load is down, and there's a paper from Korea which says that people can go to work without a negative test even, because if you have done 20 days of 14 days of quarantine and you are asymptomatic, so now that's why the guideline, which was based on real-time PCR, now is shifted to clinical symptomatology. So now they say if you're not having fever for the last 3 days, and if you're not having any clinical condition of COVID-19, and you have already fulfilled the quarantine period, you are okay even without a repeated test. So this is how the guidelines keep on changing.
Okay, now because of the lack of time, this is going to be the last question. Those questions which remained unanswered, we'll definitely assign them to the panelists and they can reply individually by mail. I think that is going to be
Definitely, yes.
the strategy right now. Now the last question is for Dr. Hafiz again. If RT-PCR positive test today and negative test tomorrow, how would you diagnose?
Very nice
That
question.
you have already explained during your presentation also. Yeah,
But
you
there is
can.
another thing which has come again now. There's a very recent thing which has come, and this is what we are facing, Dr. Godwin, and we know this. We are facing this daily. A patient who is in the late of cycle of late infection of COVID-19, and he's giving CT values less than 20, less than 30 or around 30s, which viral load is already gone down. We may be detecting maybe dead RNA. So there's a concept of subgenomic RNA. When the virus replication is such that you may be picking up some subgenomic RNA, which is not exactly the live virus, and that's why you're getting this. One sample you may get a positive fluorescence, but the other sample may be truly negative. That's why these antigen and antibody tests will literally, will give you the exact clinical picture and, of course, symptomatology. So this is what there's a need for a comprehensive approach to the whole issue, because people are paranoid, they are panicked, and they only rely on real-time PCR. But I would say, because I was a COVID patient, I can say this now. Alhamdulillah, I was good. I'm good in front of you, and there is a lot of hysteria around this. But we need to come out of it. There is a lot of the nuances as the lecture goes. There is a lot of small things in between the diagnosis, especially with respect to real-time PCR. Just so therefore relying just on one sample positive, second sample negative, and it may be, you may be in depression if you go with this. So therefore a good guidance, and congratulate Dr. Rao sir and our Rakcoms team, that we are able to contribute, we will be able to contribute through our lectures to at least reduce this paranoia in the students and the people who are hearing this.
Okay, thank you so much. We came to the end of today's session of the series of the COVID webinars. Hope all of you got very clear-cut message from today's webinar. I profusely thank our esteemed speakers for their enlightening talk. I also keep on record my sincere appreciation for the President for initiating and supporting us for this seminar wholeheartedly, our Dean for leading the way from the front, and the co-organizers, especially Dr. Chaitanya and Dr. BG, and our IT team headed by Mr. Nasir. A big thank you to all the attendees for your active participation. Thank you very much.
Thank you.
Thank you so much.
Thank you.
Thanks
Thank
to all
you.
of you.
Thank you.